Okay, everybody, welcome to the Nearpod lesson for Chapter 6 on color perception. So in this lesson, let's just look at the overview first for the chapter, and then we'll look at what we'll cover in class and what we'll cover in the Nearpod lesson. So right now we are going to be covering color detection and the trichromatic theory of color vision. And we are also going to look at the foundation for the trichromatic theory of color vision is that our color vision, our color perception is based on us having three different types of cones or color perception. So we're going to look at some of the physiological evidence for there being three receptor mechanisms, so three types of cones to help us perceive color. And we're also going to look at why we have to have three different types of receptors in order to have normal color perception. And then next in this lesson, we're going to look at some cases of individual differences in color perception, particularly types of color blindness or color deficiencies that help us understand why it's important to have three different types of cones for proper color perception. So we can certainly look at why it's important to have three cones to see color properly by looking at cases where people don't have three cones and the types of color deficiencies they have. So that's where we're going to get to in the Nearpod lesson, this one right here, and the other topics will be covered in class. So opponent process theory of color perception, looking at which theory is correct. So the trichromatic theory, the one saying that we have three types of cones, or the opponent process theory. So we've got these two different theories of color perception. And as far as which one's correct, spoiler alert, neither one of them are incorrect. It turns out that they just both explain color at different levels of the visual system. Okay, we're also going to look at color in the cortex just briefly, and we'll also look at color constancy. So color constancy is an interesting topic, and we are going to look at color constancy by looking at the dress illusion, if you're not familiar with that. Okay, so those are some of the topics that we'll be looking at between the Nearpod lesson and the live lecture. Okay, so I'm going to break here for just to give you guys a chance to watch this video. It's a quick review video on different wavelengths. So let's watch that, and then we'll move forward talking about color detection and how we discriminate different colors. Okay, so let's check this video out first. Okay, so wavelength does not always predict color. So different colors are associated with different wavelengths. That is true, but what wavelength that we perceive depends to some degree on the context that we're looking at it in. So when we look at the visible electromagnetic spectrum, it does seem that there is a correspondence between color and wavelength, right? So, different parts of the electromagnetic spectrum do seem to correspond to the colors. So this would suggest that color is a property of the light reflected from an object and therefore a property of the object itself. But we know that from at the beginning of the course that that's not exactly true, right? So Color is not a property of objects. It's not a property of, light that's reflected off of them. Color is a construct of our brain that is based on how our photoreceptors in our eyes process those different wavelengths. So there's not a one-to-one relationship between wavelength and color. And as we can see here in this demonstration that I'm going to show you, that that wavelength itself does not always predict the color that we will see. So if we look at the circle on, the square on the left and the square on the right. Okay, so square on the left, square on the right, they look like they're different colors, don't they? I think they do. I think this one looks more gray, and I think this one looks more mauve. So the circle on the left and the circle on the right do appear to be different colors, although they are actually reflecting exactly the same wavelengths. So they are, are the center squares the same color? I think they look the same color, but if we remove that background, we can see that they are actually the same color. So I was wrong. They're the same color. I didn't think they looked the same, but if you remove the background, they are indeed the same color. So the circle on the left, the square on the left appears, and the one on the right appear to be different colors, but they are reflecting the exact same wavelengths. So if you remove that background, they appear the same. So color and the wavelength that our visual system absorbs doesn't necessarily predict the color that we perceive. This is a great example of that. So the reason for this is that color is not a physical property. It's a psychophysical property. And our perception of color, our experience of color, what we're talking about is the observer's experience of wavelengths on the electromagnetic spectrum. Okay, so photoreceptors are filled with light-sensitive chemicals called photopigments. So we had talked about this briefly at the beginning of the course when we were looking at transduction and looking at, absorbing photoreceptors, absorbing photopigments, photoreceptors absorbing light and making those proteins change their shape, release energy. So we've already kind of looked at this, but we know so far that photoreceptors, they contain these light-sensitive chemicals called photopigments. So photopigments are unstable. They're unstable, and by unstable in this case, I mean that when light passes through these photopigments, they change their shape. So again, this comes back to the beginning of the class. So they change their shape in this process called isometrization. So when photopigments change their shape, this causes, an ensuing chemical reaction that ultimately results in neural responses along the visual pathway. So we have two types of receptors, right? So we've got two types of receptors that contain different kinds of photopigments. So we've got rods and we've got cones. So rods and cones, they function similarly, but their chemical contents, so the photopigments that are contained in rods and cones and their neural connections give them different specializations. So rods and cones, they have different chemical contents, and this means that they have different specializations. for perceiving or for reacting to different parts of that electromagnetic spectrum. So the rod photopigment is called rhodopsin. Now, rhodopsin is more sensitive to light. So remember, again, with our rods, that they are more sensitive to light. We get not as good a visual acuity with them, but they are more sensitive to light than cones. So rods, they contain rhodopsin. Rhodopsin is more sensitive to light than the cone photopigments are. So for this reason, rods function better in dim light than cones do. Photopsin, on the other hand, this is the pigment that is contained in cones. And photopsin, on the other hand, it requires a high level of light intensity to get these photopigments to activate. So requires a high level of energy. So your cones are basically non-functional in dim light, but they function really well in daylight. So we do have 3 varieties of photopsin. So photopsin, the photopigment contained in cones, we've got 3 types. So 3 varieties of photopsin, they're located in different cones, and they respond differently to different wavelengths of light. So this means that cones can distinguish among different wavelengths, whereas rods can only differentiate among different levels of light and dark, because there's only one type of photopigment, rhodopsin. So because cones have these 3 types of photopigments, they can 3 varieties of photopsin. These 3 varieties of photopsin allow cones to differentiate between different wavelengths, whereas rods can't because there's only one type of rhodopsin. Okay, so let's look at some theories of color vision, color perception. So we know quite a bit about cones, rods, photopigments, the chemical reactions that go on there when wavelengths of light are absorbed. So you might think, because we know all these things, in terms of different cones and what photopigments they contain, their different response properties, you might think that, clever anatomists, physiologists identified those 3 cone types and built a theory of color vision around it. This isn't the case. Research has confirmed the physiological basis of the trichromatic theory of color vision, but the basic theory itself was established by psychophysical experimentation and theorizing that actually started in the 1800s. So we know a lot now about the physiology behind our 3 different types of cones, and we know all that now, but this story started pretty well before we ever knew anything about the anatomy and the physiology, way back in the 1800s. So in the 1800s, Thomas Young proposed that color vision results from the actions of 3 different receptors. So as early as 1802, you don't need to know that a specific number, we just need to know that this is pretty early. This is quite a while ago. So way back then, Young suggested that the eye contained different photoreceptor cells that were sensitive to different wavelengths of light in that visible spectrum. So later, in around the mid-1800s, another researcher, Hermann von Helmholtz, expanded on Young's original theory, and Helmholtz suggested that the cone receptors of the eye were either short wavelength, medium wavelength, or long wavelength cones. So these different wavelengths corresponding to different colors, so short wavelengths responding to bluish colors, medium wavelengths being the greens, and long wavelengths being the reds. So Helmholtz also proposed that it was the strength of the signals detected by the receptor cells that determined how the brain interpreted color in the environment. So Helmholtz discovered that people with normal color vision needed 3 wavelengths of light to create different colors. And he did this through a series of experiments. So he conducted experiments to demonstrate that people do in fact need 3 wavelengths of light to create different colors. So in honor of, each of the contributions made by Young and Helmholtz to this trichromatic theory, it is sometimes just referred to as the Young-Helmholtz theory. So Young's contribution was not proposing that color perception is based on 3 principal colors, but he didn't do any actual experiments. So Helmholtz's contribution was really extending Young's proposal and then suggesting that there were 3 receptor mechanisms ultimately underlying color perception. So Helmholtz did the experiments, and there's also another key player in the trichromatic theory saga. So this man was named James Maxwell, and he did many additional experiments to support this the trichromatic theory. Now, unfortunately, for Maxwell, his name was lost to history as far as the theory goes. You start getting into 3 names, gets to be a bit much. So anyway, Maxwell was left out of the naming of the theory, but he did provide a lot of the behavioral evidence for trichromatic theory with his color matching experiments. Helmholtz used similar technique as this as well, but this is what Maxwell did. He provided a lot of the behavioral evidence. So an observer in Maxwell's experiments would basically try to use different amounts of 3 primary colored lights. So we've got our lights here. We've got we've got green, blue, red. So you would, if you were participating in Maxwell's experiment, you would use these 3 primary colors to exactly match another reference color. That would be this one here, the kind of bluish or cyan looking light. So what you would do in this experiment, you're going to adjust the different wavelengths of light in these 3 to try to match that reference color. So one of the key findings from Maxwell's color matching experiments was that only 3 mixing lights were necessary to match any reference light that he would give them. So 2 primary colors were not enough. So if you only had blue, green, if you only had green, red, that wouldn't be enough to match any reference color that you would be given. So 2 primary colors weren't enough. 4 would be more than was needed. But by adjusting 3 wavelengths in that comparison field here, any reference color could be matched. So having only 2 to adjust was not enough. 4 was too many. So this was a key finding from Maxwell's studies, his experiments, to show that color perception, it's based on 3 color mechanisms. So the conclusions from these experiments are that color vision depends on 3 color mechanisms. So in this case, we're talking about 3 receptor mechanisms. And each of these receptors has different sensitivities to wavelengths on that electromagnetic spectrum. So each has different spectral sensitivities. So long before physiology could prove anything, these results led Young and Helmholtz to deduce really that 3 different color mechanisms must limit the human experience of color. Okay, so then we went on to develop many more advancements in technology so that, the physiological evidence for trichromatic theory of color perception could be supported. So some of the physiological evidence is just researchers being able to measure the absorption spectra of visual pigments found in those receptors. Okay, so we're coming back to this a little bit. So we've seen this figure before in the context of transduction. We're going to come back to it again and expand on it a little bit more because color perception requires a bit of an expansion because we're now talking about 3 types of cones. So anyway, before we get to that, all visual pigments contain opsin and retinol. So opsin is that large protein here, and retinol is the molecule that comes off it. So all the visual pigments contain opsin and retinol. So opsin is this large protein here along the bottom. So the structure of the opsin protein is what determines the wavelength that a particular receptor responds to. So there are differences in the structure of this long opsin protein part of the pigments. And these differences in the structure of the opsin protein are responsible for there being 3 different absorption spectras. So we've got long, medium, and short. So cones have can have 3 different types of opsins, which respond to either long, medium, or short wavelengths. So photoreceptors also contain retinol. So retinol is that light-sensitive molecule on top of the opsin protein. And retinol captures photons of light. So retinol selectively absorbs wavelengths of light. And it is, retinol is something that is derived from vitamin A. So vitamin A is manufactured from beta carotene, and retinol is derived from vitamin A. So, this is why your mother might have told you to eat your carrots. They're good for your eyes. That is indeed true. So the opsin protein and the retinol molecules are connected. And remember from earlier in the course that retinol will change its shape when exposed to light. So it's curved now in this picture. If it's exposed to light, it will straighten up. So our photoreceptors have one of 4 types of visual pigments. So each photoreceptor has only one of 4 types of visual pigments. So one of these pigments is going to be rhodopsin. So rhodopsin, that's what rods have. And it is also a type of opsin protein. It just happens to be the one found in rods. So rhodopsin is extremely sensitive to light. So remember, this is why rods are extremely sensitive to light, and that's why they function best in dim lit conditions. It's because the type of photopigment that they have is very sensitive to light. Now, each cone has one of the other 3 pigments, which respond to either long, medium, or short wavelengths of light. Okay, So let's look at the parts of the spectrum that the different types of photopsin respond to. So cones have photopsin, and this photopsin that will respond to either short wavelengths of light in the 419 nanometer range, medium wavelengths in the 530-ish nanometer wavelength range, and long wavelengths in the 558 nanometer range. So you can think of this, although it's not completely true, because of course these wavelengths, they exist on a spectrum. So it's not absolute, but you can kind of think of the short wavelengths as being bluish, medium wavelengths as being greenish, and long wavelengths as being reddish. It's kind of a good way to remember them, keep track of them. Okay, so this picture was created by using a certain type of microscopy and some selective bleaching. And this allows for the different photopigments to be isolated. So this picture is to give you an idea of the percentages. So you can see here, we've got a lot more red going on in this picture than we do blue and green. So we have many more cones that respond to long wavelengths of light, so wavelengths of the reddish variety. We have about half as many that respond to medium wavelengths, so about 30%, responding to the greenish variety. And we really have only a few that respond to those shorter wavelengths of the bluish variety. Okay, so the retina contains 4 types of photopigments. We originally learned that there were 2 types, photoreceptors, rods and cones. That's still true. But when we start to talk about color perception, cones can just be further divided into the wavelength that they prefer. And the wavelength that they prefer is determined by the type of photopsin that they contain. So different cones, cones differ in their sensitivity to the wavelengths of light, depending on which photopsin they contain. So we've got our 3 cones, and they have maximum sensitivity at short, medium, or long wavelengths. And we've got just this one single type of rod receptor here in the middle, just remind us that there's 4, but we don't really talk too much about the rods in color perception. But we do have a single type of rod receptor, and it has its peak sensitivity between those S cones and the L cones. So the ones responding to bluish and the ones responding to greenish. We've got our rods right in the middle there. So you'll notice, though, when you look at these curves, they're kind of wide, right? I don't know if maybe you notice this, but I'll point it out. So these curves, they're pretty wide. So it's because they're wide, this means that, S cones don't only respond to, 420 nanometers. That's just their peak response. That's where their peak sensitivity is. But it doesn't mean that our S cones won't respond to, light that's down, a little bit further into that shorter end of the spectrum. And it also doesn't mean that the S cones don't respond, to a little bit of the medium part of the spectrum. So these curves are wide, and this means that each type of cone will respond to a range of different wavelengths. So S cones, they respond with peak sensitivity to 420 nanometers, but they will also respond to 400, 450, 500, 320 nanometers. And the same can be said for the other cone types as well. So we're talking about a range of responsiveness with a peak sensitivity. So just to kind of come back and remind us, so S cones, and S cones are most sensitive to shorter wavelengths of light. You can kind of remember them as being blue, although this is not entirely accurate. Because remember when I had the color coded, S cones in blue, M cones in green, L cones in red to help you remember. This is why I said it's not entirely accurate. Because S cones, they will respond to other wavelengths as well, but they just prefer those shorter ones, which happen to be in the blue range. So we also have our M cones, which have a preference for medium wavelengths. You can think of them as being roughly green. But again, this is not entirely accurate because M cones, they prefer medium wavelengths, which appear green, but they will also respond to other wavelengths as well. And lastly, we have our L cones, which are cones that are sensitive to long wavelengths of light, and long wavelengths are in the red end of the spectrum. So you can think of the L cones as being red, but again, they will respond to other wavelengths as well. Okay, so the wide range of responsiveness of the different types of cones that we have kind of creates a problem. It creates a problem for us being able to distinguish between different colors. And if I just can flip back to here, what we have is, I'm just going to try to see if I can pick out a part real quickly. So basically, what we can have is multiple cones, cones being active to a range of wavelengths, and that can make it a little bit ambiguous because they will express. Okay, sorry about that. My Collaborate just cut out. So in order to kind of get away from this ambiguity, we need to have three types of cones. So we need to have three different types of cones so that we can discriminate between different colors. So we've got one type of rod. We need three types of cones so that we can distinguish between all wavelengths, because if our cones respond to a range of wavelengths, but just some more than others, how can we tell one color from another? How do we discriminate between one wavelength and another? So we have three types of cones because that's the only way to discriminate between the different colors that we perceive. So this kind of conundrum of, how we know what colors we're looking at when each type of cone has such a wide range of the spectrum that it will respond to. So this problem is formally known as the principle of univariance. So basically, with the principle of univariance, it just means that a receptor does not know which wavelength it has absorbed. It only knows the total amount that has been absorbed. So this means that an infinite set of different wavelength combinations can bring about exactly the same response from a single type of photoreceptor. So that means one type of photoreceptor can't make color discriminations based on wavelength. So we need to have three types of cones. Having three types of cones allows our visual system to essentially use the differences in the responses of three cones and also use their relative responses to each other to determine what wavelength we are looking at. So let's bring up some graphs and kind of look at why this is a problem. So this figure right here provides a really good illustration of the problem of univariance. So this is showing the theoretical response of a cone receptor to two different wavelengths of light. So we've got 450 down here. So this is a response to 450 right here with this cone. So this is the response plotted here for the wavelength. So 450, that's here. And then we also have the response to a 625 nanometer wavelength. So if we look at the peak response up here for this cone, it's about in the 535-ish range. So this is an M cone. We're talking about an M cone that responds to those medium wavelengths of light, also appearing green. But lights of 450 nanometers and 625 nanometers have the same response, right? So if we're looking at this is the response to a 450 nanometer wavelength of light. This is the response to a 625 nanometer wavelength of light, and you'll see it's the same response. So this graph is showing that light at either 450 nanometers or 625 nanometers produces a response lower than the peak response, about that 535-ish range. And more importantly, this is showing the exact same response of this receptor to two completely different wavelengths of light. 450, 625, you've got the same response. So in this case, so we've got really an infinite set of different wavelength and intensity combinations that can elicit the same response from a single type of photoreceptor. So in this case, the response of this photoreceptor is the bluish light. And this is the same as the response of this photoreceptor to the orangish light. So we have the spectrum below to tell us where on that spectrum the light is coming from. So we have that, but our visual system doesn't have that. So the visual system doesn't have this down here. All it has is this. So it's got no reference for the wavelength. It only has the response of our cones. And if cones are showing the same response to different wavelengths of light, there's no way for us to tell what color we're looking at. So when it comes to seeing color, the output of a single type of photoreceptor is completely ambiguous. But if we have more photoreceptors, this is going to allow us to tell the difference between the response to lights of different wavelengths. So we can detect, with three types of cones, we can detect differences between wavelengths or mixtures of wavelengths with more than one type of cone. So having three types of cones allows us to tell the difference between lights of different wavelengths, because when we're talking about under brightly lit conditions where our cones are active, the S, M, and L cones, they're all going to be active. So they're all active in brightly lit conditions. Now, when they're all active, the differences in activity between these three types of cones is what makes it possible to distinguish color. So let's bring back our graph and add a couple of more cones in here and see how that improves the situation of being able to distinguish between colors that have the same, that are eliciting the same response from the cones, from a cone. So we're able to detect differences between wavelengths and mixes of colors because we have more than one type of cone receptor. So this figure shows us how our three types of cones can tell the difference between different wavelengths. So let's look at the three cone responses of these three cones to the two wavelengths of light now. So we're coming back to our 450 and our 625 nanometer wavelengths. So the responses for these three cones for 450 and 625 nanometer wavelengths. So these So this here, you know, we've got this one that was showing the exact same, this M cone showing the same response here and here to these two different wavelengths. But these wavelengths will produce different outputs from the S cones and the L cones. Right. So if we have this additional input from our S cones and our L cones, now our visual system doesn't have to rely on this ambiguous input right here. It can rely on the fact that whatever the particular wavelength of light that we're looking at, the S cone is showing a large response to this wavelength. So although the medium cone is showing the same response here as it does here, the S cone is showing a much larger response here. And then if we look at what the L cone is doing, it's showing a much smaller response to the 450 nanometers. So that's good additional information that the visual system can use. And then if we look over here to what the L cone is doing for the 625 nanometer wavelength, it's a big response here for the L cone. So we can take all of this, our visual system can take all this information, all these differences in how the three cones are responding to this one type, these two types of wavelengths that would be producing the same response in the M cones. But when we have the input from these additional cones, it makes things a little less murky. So our visual system can detect the difference between wavelengths by looking at what all three types of cones are doing. So in this case, the output of the M cones, not really all that informative, right? Because it's responding the same way to two very different wavelengths of light. But if we look at what the S cones and the L cones are doing, starts to become clearer. So the S cone response, it's large for the 420 nanometer wavelength, but the L cone response is small. So it must be a bluish light because otherwise the L cone and the S cone would response would be reversed. So cones have this problem with this principle of univariance, but that problem is solved by having three different cones. Now, all rods contain the same photopigment molecule, which is rhodopsin, and all rods have the same sensitivity to various wavelengths of light. So this means rods obey that principle of univariance. And you know, we can't sense differences in color with rods, of course. But rods do follow this principle of univariance. Okay, so before we move on to looking at individual differences in color perception, let's just kind of come back to this univariance concept. So the univariance explains, the lack of color in dimly lit scenes, right? So remember that there's only one type of rod. All rods contain that same photopigment molecule rhodopsin. So this means that they all have the same sensitivity to wavelengths. So as a result, although it is possible to tell light from dark, under dimly lit conditions, the problem of univariance makes it impossible to discriminate colors, right? So our nighttime, kind of lack of color perception at nighttime is one hint that color is indeed a psychophysical, not a physical property, right? So the world seen under a bright moon doesn't appear to be as physically drained of color. So the same mix of wavelengths that produces color perception during the day remains present, on a moonlit night. But we fail to see colors under dim illuminations, like moonlight, because dim light stimulates only the rods, and the output of that single variety of photoreceptor doesn't permit color vision, because we don't have the differences, we don't have the relative differences in rod responses. So they do obey the principle of univariance because we've only got one, but it does restrict our color perception. Okay, so we'll do a little recap video, and then we'll move on to individual differences in color perception. Okay, individual differences in the ability to discriminate colors. So perhaps you've thought of the question of, whether or not my perception of blue is the same as your perception of blue. So do we experience colors in the same way? And because color is not a physical property of the world, it's a creation of our nervous system, it's a reasonable question to ask whether or not my perception of blue is the same as your perception of blue. So does everybody see colors in the same way? Yes, in general, they do. When we're talking about basic colors, so single words that describe colors and have meanings that are agreed upon, this is our, the basic color terms that we can agree on, kind of single words that describe colors, meanings that are agreed upon. So basic color terms. Yes, turns out we can agree on these things to some degree. So let's just kind of look at this study that was done on this back in 2014. So in this study, Lindsay and Brown, they asked people to name color patches. So they presented people with all these different color patches. As you can see, they, start out more basic, and we just get more kind of nuanced in terms of the color as you go down the list. So they asked people to name color patches. Now they use these 11 basic color patches that kind of, your very basic ones that most people can agree on. So basically what we're looking at here in this figure is, the bars, the length of the bars represents the proportion of people who used these color words to describe the color patch. So what they did, they gave 50, so there's 50 people in the study. They gave them about 330 color swatches to label. And what they found was that basically everyone in the study used these 11 basic colors. So, so everyone, you show someone a black, a white, red, yellow, green, blue, brown, pink, orange, purple, and gray. Pretty much everybody would label these color swatches in the same way. But once you start getting down into these more nuanced, color patches, you start to see a very gradual reduction in people who were using those labels. So you can imagine, if someone is an interior designer or an artist, they're going to be more likely to use, labels for these types of labels for these more nuanced colors. So your lavender, lilac, periwinkle, magenta, right? So these are some pretty nuanced colors. And I often think about, changing paint colors in my house, and asking for my husband's input on it is basically pointless because he would be in this 11 basic colors. Something is brown or it is white. There'd be, like maybe just going down through here, maybe you want something tan. It's not quite brown, but it's in that realm. Greens are another good example, right? So maybe you want forest green versus more of like a lime green. So yeah, no point in asking my husband to pick colors because he's got these 11 basic ones. Anything else is just one of those. So not everybody, experiences these nuanced colors and can, put a label with them. So not everybody sees all colors in the same way. There's some agreement when it comes to the basic ones. But what about people who are, legitimately have a legitimate case of color blindness? So small part of the population does experience some form of color blindness. It turns out to be about 8% of the male population and about 0.5% of the female population that has some form of color blindness. So if we think about how important it is to have 3 cones, we can kind of see why people with color blindness have difficulty discriminating colors. So we are relying on the relative activity of 3 cones to discriminate colors. And the problem is that people with color blindness, they don't have all 3 cones, which means they're going to have difficulty distinguishing between colors. So sometimes the term color blindness is thrown around. So it's used, but people who experience color blindness, they are not truly colorblind. They can still make some discriminations based on wavelength. So they're sometimes just referred to as being color anomalous because most people who are colorblind can still make some discriminations based on wavelength. Okay, so let's look at the different types of color sight that we can get with what cones we may or may not have. So starting with trichromats, this is normal color perception, people who have 3 types of pigments, meaning that they can discriminate among more wavelengths across that spectrum. So this is what we've been looking at normal. Okay, sorry about that. It appears that Collaborate is giving me some difficulties. Okay, let's get the slide back up. Starting with dichromat.